which become less active when you're doing a task, and it seems that they're more active when you're just sort of hanging out. Okay? So that's why they call it the default mode network. You know, default meaning this is what you do before the experimenter tells you to do something. And there's lots and lots and lots of studies on this these days, and then it's become very, very interwoven for a couple of reasons with work on resting state connectivity. Well, for the most obvious reason is because when someone's doing resting state, they're just hanging out. Okay, so the things which really are active, okay, are this sort of like default mode network. And if you just get people to hang out in the scanner and look at what's correlated with what, then it turns out that pretty reliably, this has been replicated by lots of labs, lots of different times. All of these sort of default mode type areas tend to correlate with each other. And then there's this other area that seems to be more maybe like attention type areas, maybe so like a bit higher up in parietal cortex, this sort of like prefrontal, dorsolateral prefrontal sort of area, some more kind of mid-level vision areas. They seem to be sort of anti-correlated. So the blue stuff goes up and the yellow stuff goes down, and vice versa. And if you plot a correlation of, so this is a correlation matrix, the degree to which different areas are correlated with each other positively or negatively, just by eyeballing it, you can kind of sort of see that it sort of splits up a bit. So all the default areas, the areas that are kind of thought of as being part of this default network, like this, they're all up here, and they're all quite, you see there's a lot of like bright stuff here. That means they're quite correlated with each other. And there's a lot of blue stuff out here, like bright blue means they're quite anti-correlated with the other things. Okay? And you can see that some other areas tend to be correlated with each other too. Okay, so motor areas tend to be correlated with other motor areas. Visual areas tend to be correlated with other visual areas. Okay, so this is to say this is a very active area of research is an understatement. This is probably the most active area of research in fMRI right now. And people, one of the things that people have looked at is, can we sort of fractionate this out a little bit? So it's not, I mentioned already that, areas of particular modality tend to talk to each other. So it's not just sort of, the default mode and then some anti-correlated other stuff. But if you try and break this down a little bit, then there's multiple different systems that seem to, when you're just hanging out, seem to sort of talk to each other a lot. So visual I already mentioned, is visual areas tend to talk to each other. Motor areas tend to talk to each other. These are called control areas. You would call this, prefrontal, attentional, self-regulation sort of stuff. And parietal is often involved in that. Here's that default mode stuff again. Salience. I mean, these are labels. These areas are certainly, and Buckner would be the first to admit, are involved in many things. But if you're going to give a one-word area label to this, it's not crazy to call it salience. Like parietal cortex is definitely involved in detection of attentional responding to salient events. Okay, you can see there's some overlap here with this dorsal attention network. Maybe these are really the same thing. I don't know. Okay, so there seem to be multiple different areas that just without really being pushed from the outside seem to be talking to each other, or at least seem to be correlated with each other. Now here's an example where, if they're correlated with each other, you can say with some confidence it's not because they're just being driven by the external task, because there is no external task. Okay? But they might be being driven by some internal task. I mean, one of the great things, and people talk about this a lot, everything to do with this people talk about a lot. Okay? But, what is going on in people's heads when you just ask them to hang out in the scanner, right? What do you tend to think about when what do you think about when you're in the shower? Well, you might think about, well, what am I going to have for dinner tonight? What do I need to do right now? One thing that people tend to think about is, I was just daydreaming, I was thinking about that, I saw this nice movie last week, and you might sort of just reminisce and recollect things, or you might sort of think about what you're going to do in the future. There's a whole theory, which actually has some evidence for it, that these default brain areas are very involved in sort of like remembering the past and thinking about the future. So memory research, and Randy Buckner also is another person he's done a lot of this work, is sort of interested in that as well. So Now, the Turk-Brown article sort of talks about this a little bit. It says, well, it's important to bear in mind that there's changes in connectivity that happen when you're actually doing a task. Here's a modulation of connection depending on whether you're attending to faces or houses. And I think, I'm all in favor of this is just my personal opinion that you should not regard as definitive, but everyone has different views on this. But I think default mode stuff and resting state stuff is great, but it's not entirely clear. I think it's always worth asking the question, how about doing a task? Okay. So here's just a metaphor from a different area. So it turns out it is actually the case that if you look at people's resting state heart rate, you just have them sitting there in an armchair. People who are really top athletes have lower heart rates. I guess it's because their hearts are like super efficient or something. Okay? So your average person who's not especially athletic like me or something has a heart rate, I don't know what the average heart rate is, 85 beats a minute or something. People used to give the example of Lance Armstrong. I guess they don't give this example anymore. But if you take like a really, really healthy person, they might have a resting state heart rate of 60 beats a minute. So I don't know what the numbers are, but it's lower. Okay? So if you want to say, I want to see how healthy and athletic someone is, looking at their resting state heart rate is not a terrible measure. But if you go to the doctor's office and you say, doctor, I'm worried that I've got a heart condition, he's like, okay, let me hook you up to this EKG machine. I'm going to put you on a treadmill, and I want you to like jog as fast as you can. And he's going to see what happens to your heart rate and how that relates to your jogging speed. You can see the relation to like a task, right? You give someone a task, you measure the behavior on the task, how fast they're running, and you look at the response. Okay? And that's a very, very informative measure of how healthy is someone's heart. Okay? And that's what the doctor will do, I think. Okay? And in my view, which is this my personal view, although it's the case that you could get a lot of information from someone's resting state heart rate, why not do this? Okay? Now, I already said there's all kinds of good reasons for looking at resting state stuff. You don't have to worry about the task. You can combine data across multiple labs. It has interesting relations to this default mode area. There's all kinds of interesting stuff. And, so anyway, this is just something to think about. Okay, so those are some types of connections looking at functional connectivity, where functional, a more clear label would be how correlated are things with each other connectivity. Okay? But there's also structural connectivity, and it turns out that you can measure this with MRI as well. Sort of indirectly, but not that indirectly. And that's this thing that previously there was just this unexplained abbreviation, DTI. It stands for diffusion tensor imaging. I don't think Jean-Louis mentioned this in his previous class. Okay, well, great. What's diffusion tensor imaging? Okay, so it turns out, and I'll try to sort of explain why it's the case. It turns out that you can actually get a pretty good look at how strong are someone's white matter connections using MRI. Now remember I said that white matter is these sort of long axonal fiber bundles that are covered in this sort of insulating sheath called myelin. So myelin is not just insulating against electrical, the ionized currents going in and out of it, but it's also just, kind of an impenetrable lump of fatty stuff. Like water doesn't go through it either. Okay? So if you have, these are meant to be cross sections of a bunch of different axon bundles. Okay? So if you have a really tight axon bundle with really big, thick walls around them, really, myelin walls, then there's only these little tiny gaps between them. Okay? So if you're a water molecule sort of stuck either inside one of these axons or between one of these axons, you can't really just diffuse in any old random direction for very long because you're going to bump into a wall. Okay? So the most dominant direction of diffusion is going to be along the axis of the bundles. Whereas if you have a bunch of loose bundles that don't really have any myelin sheathing around them, the water can go anywhere it wants. Okay? So the jargon term for this is anisotropy, okay? Fractional anisotropy. So isotropic means same in every direction, anisotropic means different in different directions, and fractional means the proportion of time is different in different directions. So the jargon term means, if you were to plot a sort of sphere or stretched out ellipse indicating which way does the water tend to diffuse, the more stretched out it is, the higher the fractional anisotropy. Okay? Now, then this raises the interesting question, how on earth is it the case that you can actually measure the diffusion of water with MRI? I thought we were measuring, blood oxygenation level. Okay, so I'm not an MRI physicist, but I'm going to try and explain that a little bit. So, remember in John Lee's lecture, he said that the thing that you're really measuring is how uniform is the signal. Okay? So, in the MRI, it sends out these pulses that sort of tip all the spins, and all the spins start spinning. They start spinning at a rate that's proportional to the magnetic field. So if you have a really, really uniform magnetic field, all the spins are going to go around at the same rate. Okay? But these spins go around super fast. So if you only have to have a tiny, tiny, tiny difference in the magnetic field, and pretty soon, one spin, if it's even going, 1/1,000,000th of a percent faster than the spin next to it, it's going to get out of sync with it really quickly. Okay? Because they're literally, I don't even know what the rate is, but it's many millions of times a second. Okay? So you only need a tiny, tiny irregularity in the magnetic field, and it starts to make the signal go down because things start to go out of phase with each other. Okay? And that's what you can measure. Now that's actually what the blood oxygenation measure, BOLD is measuring as well, because in that case the thing that makes the magnetic field less uniform is the fact that your blood, when it has oxygen in it, all of a sudden it's like all the hemoglobin molecules become like little magnets. And all these little magnets that are not oriented or floating around make the signal less uniform. Sorry, it's the deoxygenated blood, which is like the little magnets. Okay, so the more deoxygenated blood you have, the less uniform the magnetic field is, and so the signal goes down. Okay? Well, another way for the signal to go down is for you to, you send out your little pulse to make all of the hydrogen nuclei in the water all spin around at the same rate, and the rate at which the magnetic field changes, this is called the field gradient, it changes as a function of distance. You set that up, like the scanner sets that up. Okay? And the stronger the field, the faster the things will spin around. Okay? So if you have something, right here, all of the molecules are going to spin around at this speed. But if you go over, 1/10 of a millimeter to the right, along the gradient, they're going to spin just a tiny bit faster. Okay? So suppose you have a water molecule and you may start it spinning over here, and it just starts to drift a little bit. Okay? It drifts a little bit over to where its new neighbors are spinning at a slightly different rate. And that makes the field less uniform. Okay? So that's how you can measure diffusion. That's a sort of non-physicist explanation of how you can measure diffusion. Because for a long time it was very mysterious to me. How on earth can you measure water diffusion with MRI? It's because of that. So you make the water, you get the water, it's really the hydrogen part of the water that does it. You get the spin in the hydrogen to go around a particular rate based on position in the gradient, and then it drifts a little bit. And if it happens to drift along the gradient, it's going to be found itself amongst neighbors that are spinning at a different rate, and now you've got all these different spins that don't line up anymore, and now the signal goes down. Okay, so for any given direction, so you put the gradient in different directions, and you can say, well, how much is the water diffusing in this direction? How much is it diffusing in that direction? How much is it diffusing in that direction? So if you do that for enough directions, pretty soon you've got a measure of how much is it diffusing altogether. And then you end up with a picture that's sort of like this. Now, can you see this? That there's like these little, I think you can see this. There's all of these little sort of oriented ellipsoids. Okay, so every single voxel here is filled with one of these things. Okay? So the more stretchy it is, the greater the degree to which the water seems all to be diffusing in that stretched out direction. And then a standard and quite nice way of visualizing it is to sort of color code it so that different directions get colored in different ways. So horizontal gets colored in red, and this direction gets colored in sort of green. Okay? So what you can see here, and this happens to be a known important white matter tract. Okay, so I think this is probably like the anterior commissure or something like that. Okay, so this is sort of proof of concept. So we know that there's actually a whole oriented fiber bundle here, And our diffusion measure seems to be sort of working because it's actually telling us, yeah, over here the water's mostly diffusing this way, and over here it's mostly diffusing this way, and over here it's mostly diffusing that way. Okay, so that's nice. So you say, okay, this thing sort of works. And then you might say, well, I don't just want to have this, and this is very pretty, but I don't just want to know, how is water diffusing here, how is it diffusing here. I mean, just visually, and in fact, obviously they drew in this white line here, but just visually, you can kind of sort of see how to connect the dots here, right? It's connecting the oriented dots. Okay? And you can do that in a slightly more rigorous way, that you can sort of have some random walk that, with the probability of how stretched out a particular voxel is, it will walk in that direction with that probability, and then you can see, if I start randomly walking over here, how often do I end up over here? Right? And so that gives you a way of measuring the strength of connections between different areas. And this is called tractography, which literally means drawing tracts, okay? And you've probably seen this sort of like very pretty picture. You've seen this kind of picture before. This is actually, of all the pretty pictures in MRI, this is probably the prettiest kind, and these get reproduced a lot for precisely that reason. And they're very pretty. It's always worth, it's always worth asking, and this again, I'm just spouting my own personal opinion here. It's always worth asking, okay, it's pretty, but what does it actually tell us? And so we'll talk about that in a minute. Okay? But just to see what this is, okay, this is using a very similar sort of color coding, that the direction in which a fiber is going, it gets colored in a different color. So you see here, there's like I think this may actually even, it's from the same website, it may even be using the same color scheme. So this sort of like horizontal bit here is colored in red, and the sort of, 45-degree bits are colored in green. And so the strongest bunch of white matter connections that you have in your head in this thing, the corpus callosum, which joins the two hemispheres. So, if people really want to show like a really strong, nice, pretty tractography picture, they'll usually show the fibers around the corpus callosum. And so that's what's going on here. So there's, in this middle part of your head connecting two hemispheres, there's lots and lots of sort of horizontal fibers going from left to right, right to left. So that's all this kind of red stuff here. And then once they've gone out, they sort of curl around and go in all different directions to all the different brain regions. So this is showing the white matter tracks going out, far into the edge of the gray matter. This is just showing some of the tracks, but it's showing a lot of them. And they all sort of, converge through to the corpus callosum, sort of go horizontally, go through to the other hemisphere and come out the other side. And people have done validation studies on this, where you can actually take a post-mortem brain and look at the connections and then do the diffusion and see if they line up, and they do sort of line up, so that's good. Okay, so probably the only area, let the record reflect, I'm now just about to state my own personal opinions that other people are not obliged to agree with. Okay. So probably the only area of fMRI, MRI, DTI that's even more popular, but is actually related to resting state connectivity, is mapping out all these connections. Well, what's the relation? Well, you would think, there's a clear connection, which is that if you think that two areas are functionally connected, meaning they're correlated with each other, one would hope that there'd actually be an anatomical connection joining them. Maybe not a single direct connection, but at least a pathway. Maybe you have to go along this path a bit, and then over a bit, and then out through another path. Okay, so it's an interesting question to see, do the connections that we find through this fMRI measure, looking at time courses, how well do they match up with these connections that we find by tracing traffic diffusion imaging, the anatomical connections. And so this is often called connectomics. And there's lots of, this is very popular these days, and quite a lot of money being given to it. This is my opinion. Okay? There's lots of things these days called like omics. Okay, just as there's lots of things called big data. In fact, big data and omics tend to go hand in hand. And so this is a sort of like a skeptical take on it, but maybe not that skeptical. Okay, so suppose I've measured, you know, all of these things. Well, I've definitely made a very pretty picture. But, you know, one of the things I've been trying to hammer on about it, and I would recommend, you know, I think this would also be a good thing to sort of think about in your project is, you know, this thing we're measuring, is this actually really telling us anything extra about how the brain works? And by that I mean, you know, the brain's job is to keep us alive in this big, unpredictable, changing, complicated world out there. Okay? So if you're measuring something that even in a tiny way helps to figure out, you know, how on earth does the brain manage to do that, then, you know, it's clearly relating to an important neuroscience question. If you're finding something that's, you know, very beautiful picture of the structure of the brain, but it's less clear how that actually, you know, tells you how the brain is succeeding in helping us deal with the world, then it's an open question, you know, have you actually taught us anything about the brain, or have you just taught us something interesting about, you know, anatomical structures or something like that? So if you have a great tool for measuring stuff, it's very tempting to say, let's measure all the stuff. Okay? And when you measure all the stuff, you're doing omics. So if you're measuring all the connections, or as many as you can, now you saw that wiring diagram before, you can't measure all the connections, but you can measure a lot of them, okay? If you're measuring all the connections, then you're doing connectomics, okay? And that's quite popular these days. But, so here's, you know, this is slightly tongue-in-cheek, but not entirely. You know, just because you're measuring a lot of stuff, well, science does involve measuring stuff, but it doesn't mean that if you measure enough stuff, you must be doing great science. So you know, the first omics was the human genome, and everybody said, hey, once we've mapped the human genome, we'll be able to cure all these diseases. And then after what actually happened was they were like, oh, we've mapped the human genome, but it turns out that we still don't know how to cure these diseases because lots of genes are working together in really complicated ways, and now we have to try and understand that. And so, you know, if you want an example of lots of things working together in complicated ways, well, how about the brain? Okay. Well, then you say, well, that's exactly why we're looking at all the connections in the brain, because if we knew all the connections in the brain, then we'd know how all these things are, you know, talking to each other, right? Well, maybe. Okay, but remember how the connection between a given area, two given areas, is highly variable as a function of what task you're doing. Okay, so if you're doing a face-related task, the face area is talking a lot with V4. If you're doing a house-related task, the house area is, I don't like to turn the house area to the face area, but you know what I'm saying, the hotspots are talking a lot with V4. You know, one thing that definitely doesn't change in a task-related way is the anatomical connections. Actually, these might change with a function of training, and they certainly change as a function of development, but they can't change on a minute-by-minute basis as you switch from one task to another. So you can't just look at the structure of something and read out the function from it. Even if, you know, the god of neuroscience came down and actually gave someone a huge hard drive consisting of all of the connections of every area to every other area, it still wouldn't actually tell us, you know, what they're all doing. I mean, it might be useful to know, but it wouldn't tell us what they're all doing. And there's a very nice, and some people have written, this person called Corey Bargman, who's she's a big neuroscience at Rockefeller. She's written very nicely about how does a worm called C. elegans, which people have known the entire connectome of for a couple of decades now, and they still don't know how, and it only has like about 300 neurons, and they still don't know how the neurons are actually working, how they're making C. elegans survive in this big, dangerous, complicated world of ours, but they've known the connectome for 2 decades. So this is a sort of slightly skeptical take on omics. But having said that, you know, clearly if you just say, hey, we're going to measure stuff so then we'll understand everything, I mean, that's obviously not fair. I mean, most, maybe 1 or 2, but most connectomics people are not doing that. Okay? Most connectomics people are saying, you know, we actually have some quite well-informed hypotheses about how particular areas might be talking to each other, how that might be affected in disease, how that might be affected during development, and then we're going to go and look at stuff to do with that. And I'm all in favor of that, but this is just my opinion. But the mere act in itself of measuring stuff may or may not be interesting scientifically. So this might be something to think about for your project. You know, you're going to say, this is the why should you care bit, okay? So you're going to say, you know, I'm really interested in responses of singular cortex during self-control task or something like that. Well, that's great, but you have to explain to someone why you're interested in that. Okay, now there's perfectly good reasons to be interested in that, but you just have to say what they are. Okay? And that's not just my personal preference. That's true for like any time you ever communicate your scientific idea to anybody. Unless they're your advisor who already knows exactly what you're doing and why you're doing it. And even then, it's still worth explaining like what the bigger picture is. So cross out beware of omics with if you're going to measure something, say why you're measuring it, okay, and say what you're going to learn from measuring it. And the outcome ideally should be more than just and then after we've measured it, we'll know how big it is or how long it is or how connected it is or what color to color it in or something like that. But anyway, so those are some different ways of thinking of connectivity. So have a good weekend. I will write back and set up meetings, especially now I'm trying with those of you who've already pinged me. Do not feel shy about pinging me again, and if you've already pinged me a couple of times, ping me another time. Okay? I'm sorry that I require so many pings. But okay, thanks anyway. Oh, and I can also meet with you now if you want for like 15, 20 minutes. Yeah, you want to chat now? We can if you want.
Or maybe tomorrow. Yeah.
I will get back to you on that. Maybe. Let me actually look at that right now because I've already sent you a number. I mean, how was your conference, by the way?
It was okay. It was kind of poorly organized.
Oh, really?
Yeah.
Oh, yeah. Where was it? Oh, okay. Well, these things are nice places.
No, that's
Okay. How many people are there? Are we talking hundreds of thousands?
Yeah.
Oh, okay.
That's okay.
Okay. So,
Yeah, it's supposed to be 70.
yeah, I think.
Okay, that sounds good. Yeah.
Oh, you missed it. Oh, okay.
Are you going to